That would be super nice. Okay, I will continue again. Hello everyone, I'm Shoupeng. I'm very happy to be here to share with you our work, which is mainly about engineering cucurbit-like polymersomes with the aggregation-induced emission, while I also showed with the enzyme-mediated motility. So let's start with talking something about the motion and the motors. So the autonomous motion found in nature are able to perform sophisticated work. This has inspired scientific researchers to fabricate man-made motors. Due to the triggered or self-propelled motions, these man-made motors are able to and have already proved to be more efficient vehicles in the biomedical field, in the biomedical science field, compared with those compared with those passively delivered vehicles, that these man-made motors showed an enhanced efficiency in the intracellular delivery, as well as a more pronounced tissue penetration profile, which is quite important and essential to be used in the more efficient cancer killing, especially in vivo process. And in most of cases, that these synthetic man-made models are their design are based on the particles that with intrinsically asymmetrical shape, which allow the higher net force on one side, and this is essential to propel their motion. And until now, that there are various sources have been used to propel the motion. And for instance, like light-induced photothermal phoresis, as well as like the magnetic force-induced precise guide. And in addition, that besides that, the fuel-induced catalytic pathways, for instance, here platinum particles that decompose the hydrogen peroxide into water and gas bubbles, as well as the enzyme-mediated enhanced diffusion are also known as powerful approaches to induce the motors autonomous motion. Among these very nice platform that has been used for the nanomotor application, that and the polymersomes, also known as polymer vesicles, draw our much attention. The polymersomes is a typically shell as a structure that contains an aqueous and inner compartment that is enclosed by a hydrophobic bilayer membrane, which is self-assembled from the amphiphilic block called polymers. Within such architecture, that polymersomes enable loading both the hydrophilic cargos in the lumen as well as the hydrophobic drugs and in their hydrophobic membrane pocket. So this has enabled them widely used in the biomedical field. Besides to that, the polymersomes are also known to have the ability to adapt under environmental changes and undergo controlled shape transformation. So this is mainly associated with like the polymer composition, like the fabrication method, as well as the environmental conditions. And as can be seen here, a series of various of the non-spherical polymersomes like nanotubes or the polymersome domicile has been propelled using so-called osmotic pressure induced shape transformation pathways. This is induced by deflection of the inner volume of the polymersomes and membrane indentation. So so this is in details realized by dialysis of the polymersome in an organic and water solution against a salt water solution. This osmotic pressure induced formation, as well as the membranes spontaneously curvature, and subsequently kinetic trapping allows people to fabricate polymersomes that with tailored shape and morphology. And besides taking advantage of this unique morphology of the polymersome as domicile, they are also being used to propel nanomodels. This is realized by like entrapment of the catalytic species in the cavity of the polymersome domicile during the shape changing process. The presence of fuels like the hydrogen peroxide can be decomposed into water and gas bubbles, which can be released from the small holes of the polymersome domicile. That's effectively drive their motion. And also that this catalytic species can be also switched from the metal to the enzymes. And again, that the presence of fuse and enzyme cascade enable the production of the gas bubbles that will effectively enable the motion of those nanomodules like a small nano rocket. Also, these nanomodules also have showed a nice biometric use. For instance, as I mentioned before, that the hydrogen peroxide at times used as fuel to propel the motion. So this can in turn, however, that can save the cells from hours stress. So as we show here, that by incubating the cells together with the motor and with the high concentration of the hydrogen peroxide, can effectively reduce the intracellular level compared with the control group. So this can significantly increase their cell viabilities, even though when exposed to a very high concentration of the H2O2. And besides that, the motion ability of the light-driven hormone nanomodules also can contribute to a more pronounced deeper penetration. As we saw here, for the control group that without light, the models, the particles that only be located at the edge of the tumor 3D tissue, 3D and tissue. Why, however, in the presence of light and the motion behavior can effectively penetrate into the center of the 3D spheroids, so which is quite promising to be used in a real in vivo test, especially to kill a real tumor. Although there are some nicely progress towards the formation of the functional nanomodules, but we still have several drawbacks. For instance, especially when conventional drugs or dyes are incorporated in this nanomodule based delivery. Sorry. For instance, that there are the cargo leakage and the dye bleaching as well as pie stacking in just quenching that effect, that strongly hinder their therapeutic performance, especially in vivo, as well as not able to realize very efficient long-term tracking. So one emerging reason line to solve the issue is to integrate a special class of the dye which shows the aggregation-induced emission. So unlike the normal fluorescein that which show emission in the soluble state, this, but however, quenching the emission in the solid state, the dye that vary this aggregation-induced emission components that behave in a reverse way, which means that they don't show emission in the solution state in the large amount of the DMSO or in the THF. Why, however, with the increased the portion of the water, which leads them simply aggregation or self-assemble, that they can boost their emission. This is because of the restriction of the intramolecular rotation, as well as the reduced non-radiative decay that boosted their emission in the self-assembled state. The concept of the AIE has already been widely used, like in the biometric field, to propel by propelling these fluorescent particles. This AI concept can further contribute to a boost the performance in the, for instance, in the bioimaging or in the cancer therapy. Not long ago, we also tried to integrate this AI design in the block polymers and fabricate AI polymersomes. This is realized by tagging of the AI components group in the block copolymer in the end of the chain. And now some of the block copolymers don't show emission at all. However, self-assembled when self-assembled to form the polymersomes, they show much more strong and boost emission, because they show typically an aggregation-induced emission behavior. Besides that, the positive charge of the pyridine group also enable very strong interaction of the AIE polymersomes with common cell mitochondria. This is due to the strong electrostatic interaction between this positive charged surface with the mitochondrial membrane, and this can further drive integration of conventional photosynthesizer that is loaded in the hydrophobic domain of this AIE polymersomes. The mitochondrial targeting delivery of this photosynthesizer and PDT can lead to effectively cancer cell therapy, both in vitro and in vivo. although we don't see the mice. Yeah. We also tried to integrate of the AI concept in the nanomodel design. So here we used a more rigid block copolymer, which can stand of the spark cooling process. Here is we coated the polymersomes with half-cut gold, and the gold coating is verified by the TM. Because of the presence of gold, that under 2 photon light, it can produce heat. The heat effect can cause thermal fluorescence effect, thereby effect to drive their motion. This is verified by the calculation of their mean square displacement values. As you can see here, that the mean square displacement values increased with the power of the laser increase, which is a proof of the light driving motion. Combining the photosensitization ability of this AIE polymersomes, which can produce RS upon the 2 photon light, together with the photosome heating from the gold. So we're integrated with cells that these AIE polymersomes nanomodels can very efficiently kill cancer cells in a special controlled manner. Although that we have nice progress about the functional polymersome nanomodel fabrication, there are still several drawbacks we have to face with. So one thing that we have here, in this case, we have to use the light as the trigger. But in some certain applications, that the naturally available fuel is better and is more favorable to use. In addition, that because here we have to use the gold coating process. For one thing, this will need additional processing steps, which will strongly limit their scalability. So if the AIE polymersome itself can have some certain shape change and can load some enzyme by certain means, then it will be even better. So hereby we aim to using such a design to engineer an enzyme-powered AIE nanomodel. This can be realized by controlled shape transformation of this AIE polymersomes into specific shape, into specific asymmetric shape, and followed by the supermolecular layer by layer assembly, during which we can load an enzyme within this supermolecular polymersomes, and then the presence of fuels can effectively drive their motion. The polymers are synthesized via 2 steps. The first step is about the ring opening polymerization, starting from the pack 2K with the pentafluorphenol incorporated TMC trimethylene and carbonate. With a higher than 95% conversion, followed by a post-polymerization with the amino tagged AIE compounds. We are allowed with a quantitative conversion. We are allowed us to tailor the synthesis of the block copolymer with controlled AIE group, like 510 or 5H or 40 and 22. And for the GBC measurement confirmed that the block copolymer has a very narrow PDI and a very high content of the AIE, which is normally very hard to achieve via the conventional physical loading approaches. The self-assembly of the polymersome is then realized by the solvent switch method, in which we firstly dissolve the polymer into HF, and then we slowly add water, and which will drive the self-assembly, and then followed by a simple dialysis recovery process can get very nicely spherical polymersomes. Although in this case that dialysis against water and at the room temperature will give us spherical particles, but simply varying the dialysis conditions will allow us to precisely engineering the shape of this AIE polymersomes. For instance, here with a folding AIE group, that the dialysis against the molecule at 4 degree did not impact their morphology. However, that only in the presence of 50 millimole sodium chloride, we can clearly see that the shape and transformation into the tubes. If we decrease a little bit of the chain length, we can still see that the shape changes from the spherical particles into some kind of peanut-like morphology. What is very interesting is that if we further decrease the chain length to AI5, you can see that we can clearly see the shape change and engineered into a very unique cucurbit-like morphology. So the cucurbit-like morphology has also been further verified by the cryo electron tomography and 3D reconstruction. As we saw here, that the structure within this cucurbit polymersomes, which means that they are not like three small polymersomes adhere to each other. They are in fact one single integrated cucurbit polymersomes. So this provides the basis for the future research. And then we also investigated its photoemissive behavior. So the photoemissive behavior is like is verified by measuring the fluorescence intensity in the mixture of the DMSO and water. As you can clearly see here that in the large portion of the DMSO, the polymer dissolved, and thus it's going to show emission. While with the increased amount of water, that the emission has a strong boost emission, which is because of the sample induced emission. Although when we further increase the water amount, it shows a slightly decrease of the emission. This is because of the pipette stack quenching that slightly quench the emission, but that's okay. So, and also that the photoemissive nature of this canal of these AIE components are further confirmed by the confocal. So, which means that they are highly promising to be used in the biotracking. And then, since now that we have the asymmetrical polymersomes, but how can we engineer them into with the analoading and fabricate them into nano models? Because in the private case, that the analoading and in most cases are achieved using the so-called polymer stomach site, but that is not the case in this current design. So to enable analoading, we here adopt an alternative approach. Means that we here use a super molecular layer by layer assembly. Surface potential measurement confirms that we have 5 layers of PAH and PAPSS touched on the surface, during which that the enzymes like catalase and urease can be loaded within the supramolecular polymerase via strong electrostatic interactions. And one thing worth to notice is that in the case of spherical particles, it seems has seen the more analoading. This is possible because that in the case of the spherical particles, they have a larger surface volume. And the further SAM and DLS as well as acquired here measurement to confirm that with layer by layer sample did not impact the cucurbit morphology. With the unloading, that the size and cucurbit shape are still maintained well. So this build the base for the future for the further management. Now that we have the enzyme loaded like asymmetrical polymersomes, so the following study is to investigate how it behaves, how about their motion ability. So now here we see that without urea as a control, that both spherical and cucurbit-like polymersomes that is loaded with urea and behaves so a similar tracking path, and also only shows sporadic motion. But however, that the presence of urea that have a significant effect on their motion behavior, as we saw here, that in the case of the cucurbit-like polymersomes, they traveled much more significantly longer than the case than we saw in the case of the spherical particles, as well as we have got a high diffusion efficiency when we feed the urea with this cucurbit polymersomes compared with the spherical counterpart. So next, we also further analyze the motion behavior via mean square displacement values. As we saw here, that the mean square displacement values, yes, from the cucurbit-like polymersomes in the presence of urea, they increased with the increased concentration of the urea. But however, that in the case of the spherical counterparts, they more or less behave like a linear fitting. So we can start in this case that in the cucurbit shape, because of their specific asymmetrical nature, that the enzyme-mediated catalytic reaction allows a higher net force, which can effectively drive their motion. While in the case of the spherical counterparts, because they are more or less symmetrical, so they show a little bit enhanced diffusion. We then next evaluated their biocompatibility. So as we showed here, that the HeLa cells incubated with cucurbit polymersomes and cucurbit the polymersomes that with loading, in both cases they show a high cell viability over like 80%. So this is a good sign of their good biocompatibility, which also verifying their potential use in the further studies, especially like in the Biometrics. So hereby that we in conclusion that we have the engineered fluorescent polymersomes with the aggregation-induced emission and with controlled morphology. This is which is dependent on the length of the AIE group as well as the environmental conditions. One of which of the cucurbit polymersomes can be further used for the used with the supermolecular layer by layer fabrication, and upon the which can load an enzyme upon the filling with the fuel can effectively induce their motion. So we think that this nano-model design, like which integrates the inherent AIE AIE emission, as well as the vascular nature of the scaffold, as well as the enzyme powered motility, can be of course be further used in the bio-related field. So hereby comes to the end of my talk. So I would like to thank my promoter Yang and my co-promoter Dave and Lai, and special thanks to Jinxin, my close collaborator, as well as many thanks to Hang for the quality. And thank you for attention. If you have any question, I'll be very happy to handle that.